Hello, everybody. Thank you all so much for coming. It's wonderful to see so many familiar faces in the crowd here, a number of partners, clients, and then a number of new faces. This is a really special moment for me because I'm actually originally from Andover, Massachusetts, and so I'm a native. So when the opportunity came up, we were talking about opening an office on the East Coast and looking at different options for it to be Massachusetts, and in the Boston area was certainly near and dear to my heart. It's just a really exciting time for us, and so thanks for coming out to share it with us. So just a little background for some of you on SDC. SDC was started in 2005. We're about 75 people now, 12 years old. We've done over 300 trials in a lot of different therapeutic areas, are continuing to grow and make momentum forward. You know, we focus in biostatistics and data management. We work on trying to do a dedicated service model that provides a high level of statistical consulting and data management expertise to work with our clients in a partnered consultative way, and so we really try to make an effort to care about what we do.
Who are you?
Dale, I'm the CEO of SDC, by the way. Richard Abelson, sorry, didn't mean to introduce myself. And this is Dale Usner, President.
Thank you, Richard. Thank you very much, everybody, for coming out to this event this evening. I'm very grateful for you guys to come out and share this with us. SDC is very passionate about statistics and data management in clinical trials, and we're really hoping to engage in the community as we are here presenting very hopefully valuable topics and engagement in educational discussions on clinical trial topics from a data services angle. So thank you for that. I also want to reintroduce Faith Kolb, who's back there. Faith is she needs no introduction. Faith is our Vice President of Biometric Operations, and she is local to this area, has over 20 years of experience in clinical trials, and we are incredibly grateful to have her both running our operations as well as our shop here in Waltham. So, Faith, thank you very much. Jim Townsend will introduce the speakers.
We realized, since Dale's one of the speakers, that it would be awkward if he introduced the speaker, so I'll fill in for him. So we're obviously very fortunate tonight to have three very distinguished speakers here, and really excited that we've got such a great turnout. Hopefully these three won't mind me mentioning that combined it looks like it's over 75 years of industry experience sitting at that table, so definitely lots of experience to share. Our first speaker is Ken Getz. Ken's going to be speaking to us about trends in protocol design, practice, and optimization. Just a little background on Ken. He's the chairman of CIS-CRP, a nonprofit that he founded to educate and raise public and patient awareness of the clinical research enterprise. He's also the director of sponsored research and associate professor at Tufts Center for the Study of Drug Development at the Tufts University School of Medicine, where he conducts research programs on drug development, management strategies and tactics, outsourcing, global investigative site and patient recruitment practices and policies. Ken was twice nominated as a Distinguished Faculty Award at Tufts. He's a well-known speaker at conferences, symposia, universities, investigative meetings, as many of you, I'm sure, are aware. Ken has published extensively in peer-reviewed journals, books, in the trade press. He holds a number of board appointments in the private and public sectors. He is currently a board member of WCG and Metrics Champion Consortium, and he's on the editorial board for Pharmaceutical Medicine and Therapeutic Innovation and Regulatory Science. He also writes a bi-monthly column nominated for a Neal Award in Applied Clinical Trials. Ken's the founder of Center Watch, a leading publisher in the clinical trials industry. He holds an MBA from J.L. Kellogg Graduate School of Business, or Graduate School of Management at Northwestern, and a bachelor's from Brandeis University here locally. with that, I'll hand it over to Ken.
Thank you, Jim. I think that introduction took as long as my entire talk, so thank you. Thank you for that. It's really my pleasure to be here, and I want to thank SDC for the invitation to share in this celebration. And I sort of have the privilege of really setting the stage and sharing with you what many might find a little bit of a depressing topic, which is overall protocol design practice. But I threw optimization in the title because that's the hopeful part of my discussion. And I think you'll see that adaptive trial designs are probably one of the most promising areas where we are optimizing design, and it's really quite a growing area of interest among the pharmaceutical and biotech industry. All of the data that I'm going to share with you has been published in peer-reviewed journals. It's not data that we've compiled. It all comes from the companies themselves. All of the data has been coded by their own protocol authors, by their own clinical teams, which makes for an incredibly labor-intensive activity. But anytime I talk about the distribution of the practice areas, you should remind yourselves that these were coded and categorized by the companies themselves. They're actually reporting on their own practice. We have not done that for them. And I'm certainly happy to share any of these papers with you just to really give you a flavor for the variation by therapeutic area, by phase. I'm going to be kind of giving you a high-level overview of some of the most common design practices and the problems that we encounter as a result. I'm also going to do something that speakers are advised never to do, and that is to begin with all the takeaways and then get into the data and the insight behind it. But I've done one thing here. I've framed the punchlines as questions for you to get you thinking, so that will help a little bit. So I'm going to talk about design practices, and I'll characterize them for you. I'm then going to link our design practices to clinical trial performance. And as a rough rule, the more complex the protocol, the poorer we perform. And we have been able to demonstrate that time and time again. If you look at our cycle times, you look at our patient recruitment and retention rates, the cost of our studies, the success rates of our studies, they're all influenced by this fundamental roadmap that we established to essentially direct our clinical teams. And then I'll get into, after we go over the impact area, I'll quickly touch on some of the most promising optimization areas, and in some cases I'll even have some data to share with you showing some of the demonstrated areas of impact. So these are essentially the 5 questions that I'll use to frame the discussion. First, are you saying that protocol complexity is bad? And the answer, the quick answer, is no. What we're saying is it's inevitable, and in fact it's actually rising, as you'll see in a minute. The real question is, how do we manage complexity more intelligently, more prudently? And what can we apply against that complexity to support the timelines that we've established and the critical efficiency requirements that we also have set in place for ourselves? Are regulatory agencies to blame for rising complexity? I'm often asked this question, and it depends on who you ask, right? If you ask the FDA, they'll tell you, they'll point fingers at industry. If you ask industry, they'll point fingers back at the agencies. I would say, as a quick answer, there's plenty of blame to go around. And we'll leave it at that until I show you the data. Tell us what specific procedures to remove. I love that question because it's impossible for me to answer that. It depends on the endpoints and objectives of the study as defined by the clinical study report, and therefore the procedures that can be removed are only those that are protocol specific. They have to be determined by each individual protocol. Why does the Tufts Center believe that clinical trials are a waste of money? The first time I presented these findings, the next day there was a report in one of the trade journals with this as the title: the Tufts Center claims that trials are a waste of money. It's the first time I ever got a call from the dean of the medical school who wanted to know what I had said that would have triggered this. It's clearly not what we're saying. What we're trying to say is that the decisions that we make have within them some excessive or non-essential activities that actually drain and require resources that could be better spent elsewhere. And I'll talk about that in more detail. Protocol amendments improve study design. Why do you recommend avoiding them? And the reason we do is because these are unplanned and unbudgeted. And when you aggregate all of the protocol amendments, which are remarkably disruptive, you realize that there are some that could have been avoided and would have represent substantial resource savings that could have been applied elsewhere. And so that's, again, part of the thinking here and the philosophy. How do we manage complexity and the byproduct of complexity more effectively? So let's look at some of the data, and these are all now aggregate average measures. I'm looking at 2 5-year periods where we have roughly 10,000 protocols that were evaluated in each of those observation periods, separated by 10 years. Total number of endpoints has nearly doubled, and I'll show you in a minute the distribution of the endpoints. Number of eligibility criteria has also risen by 60% or so. We often hear from investigative sites that they get protocols today where they believe they will not find a single patient in the community, perhaps not even on the planet, who will be eligible for the trial. Total number of procedures has also risen dramatically, and yet the number of visits and the duration of trials has actually not risen at a comparable rate, and therefore sites are performing more procedures on patients at every single visit. Total number of data points collected, that's an interesting one. We estimate that for our phase 3 pivotal trials, we're collecting nearly 1 million data points that have to be curated and cleaned. It's data that has to be managed and analyzed. So there's tremendous amount of resource and attention placed on the data alone. And then a couple of operating figures for you here. Total number of countries, as many of you know, has doubled, and the total number of investigative sites has also risen dramatically. Interestingly, the average number of patients per phase 3 protocol has actually gone down. We're actually engaging larger numbers of sites, spreading recruitment risk to each enroll a smaller number of patients, which makes good business sense, but in many respects it makes it more difficult for the site to ever achieve a level of scale and familiarity with the protocol when they're being asked to recruit 3 patients over an 11-month period. So it has the unintended consequence of making it very difficult for our sites to really efficiently conduct our studies. This is just looking at the change in our endpoints, and the good news is when you look at those time horizons that we typically still only see 1, maybe 2 primary endpoints and a comparable number roughly of key secondary endpoints. It's everything else that we're collecting, all the tertiary and exploratory and miscellaneous or supplementary secondary endpoints, and that's driven by so many factors. At the same time that we're looking to demonstrate safety and efficacy, we have so many more pressures on our protocols to demonstrate economic benefit, to gather more subjective assessments so that we can demonstrate clinically meaningful benefit as defined by the patient community. We're gathering more data on combination therapies and diagnostics that are now bundled or part of the therapy that we're evaluating. We also know that habit plays a big part. We still see many companies that will take a phase 2 protocol and just cut and paste that template into a phase 3 design and use that as the basis for the protocol design practice. And that is a behavior or a practice that is quite problematic. And also we see many companies second-guessing, anticipating what a regulatory agency or a health authority might request and baking that into the design of the study as well. So there is a real effort to mitigate and to anticipate these requests and essentially lower that sense of risk going into the conduct of the study. There are many, many other measures that I could share with you. I'll just highlight a few more, but again, it just gives you a flavor for the incredible growth that we've seen in complexity. We have 2 other measures that we use to just try to also understand burden. We have one measure called work effort, and it was adapted from the Medicare reimbursement as well as a value to the individual performing each procedure, and we found that the work effort to administer our procedures is growing at an even faster rate. So many of our procedures are even more labor intensive and require even more time. Many of our procedures are less expensive to implement today than they were in the past, like an ECG assessment, for example. But when you aggregate all of the procedures together and you think about the total cost per patient visit, the individual cost savings per procedure has not been enough to offset the incredible growth in the aggregate of the total cost to support the administration of every procedure in our protocol. So let's now relate the design characteristics to our performance, and as I mentioned to you, it's very, very intuitive, and there's a lot of data to support it, and I'll just touch on a few areas here. For one, we see that our recruitment and retention rates overall are the worst that they've been in our history as an industry. And we find in general, when we compare the cycle times for high complexity protocols to low complexity protocols, that the more complex protocols have substantially longer overall cycle times. Interestingly, that startup cycle time from protocol readiness to first patient first visit is only modestly different. And that's because our sites typically race to get that first patient in the study. It's the downstream impact of complexity where you start to see the challenge of patient retention really come into play. And that's why we almost see a doubling in the cycle time when we compare our most complex to our least complex protocols. We can relate that even further to some of the classic recruitment durations that we're measuring today. Across every therapeutic area, the planned duration of the typical clinical trial has to be doubled in order to achieve the target enrollment rate, which is absolutely remarkable today. It could be a function of poor planning, that we set very aggressive timelines that we have no hope of achieving, but we believe the complexity of our protocols is playing a role here as well. And on the right-hand side, you see a very common pattern in any large phase 3 multi-center study. Even after doubling the planned enrollment period, 10% of our sites will fail to enroll a single patient. And about 40% of our sites will under-enroll, and that's our most expensive group because now we have to monitor them. They've been activated, and so they're now, they have to be managed. And so that is really a major challenge for our industry. You may not know this, but the number of complaints filed against investigators and investigative sites for noncompliance and fraud has increased dramatically during the 2000s, really starting around 2005. In 1999 and 2000, the average number of complaints that the FDA received was 10. And we saw a tenfold increase for a variety of reasons. One, the FDA launched a toll-free number. The FDA also offered the opportunity to file a complaint anonymously online. It was also a time when Jesse Gelsinger and Ellen Roche, and there were a few other very tragic patient deaths that really created heightened sensitivity to reporting any perceived noncompliance or fraud, and that has really continued to rise, and it's stabilized around the 250 complaints a year level. The number one area of complaints is protocol noncompliance. I mentioned to you just how disruptive protocol amendments are. We see a very strong relationship between complexity and the number of amendments. The typical phase 3 protocol has 2.3 amendments. It costs about $500,000 to implement a single amendment and requires about 3 months of time to take back or suspend the study, to take back the protocol, to revise it, to approve it internally, to resubmit it to the IRB, and ultimately to reconsent our study volunteers. If we aggregate the total cost of amendments, we're looking at about $6 to $7 billion a year, unplanned, spent just to implement our protocol amendments, which is just a remarkable number. And here's the part that depresses people the most. A high percentage of amendments are implemented before the first patient has even received the first dose, which is a clear indication that we're not spending time challenging and testing and refining the feasibility of our protocols before we implement them. The number one reason for amending is to relax our eligibility criteria. So I've now shared with you some of the characteristics of protocol design practice. I've connected the dots a bit to talk about its impact on protocol or clinical trial performance, let's look at some of the optimization areas, and there are quite a few. One area where we see a lot of focus today is on the protocol authoring process itself, and there are many places that we can look for insight. These are just two that I'm listing here. One is the SPIRIT checklist, which you can freely download. It's available online. And it was created by a community of global medical writers, protocol authoring experts. Transcelerate now has a common protocol authoring template. And they basically apply the same principle, which is to create a direct connection or a line of sight between each procedure and the endpoint that it supports. And if it's not a primary or key secondary endpoint, if it's not required for regulatory compliance or a baseline assessment, then it suggests challenging whether that is a procedure that should be administered, if it can be administered a smaller number of times, or even delayed for perhaps a post-marketing study. So that's the basic principle behind these authoring templates, and a number of organizations are using them quite actively today. We also see a number of organizations, large and mid-sized pharma companies, that have implemented feasibility review committees that actually challenge a pre-approved protocol with an eye toward improving its feasibility, the ability to administer it more easily and conveniently. And many of these feasibility communities are 10, 15 years old at this point. It really depends on the culture of the company. We find that German companies, for example, call them feasibility challenge committees, whereas American and British companies often call them feasibility facilitation committees, and I'll let you think through the cultural differences there. Another way that we challenge feasibility is by actually soliciting patient input, and we see a growing number of companies now using patient advisory boards. These are patients who are not actually participating in the study, and they're given the protocol before it's been finalized, and they typically offer insights into the schedule of assessments and what it might be like, the burden of participation from their perspective. And so these are proving to be quite inexpensive to implement and quite valuable in terms of the insights that they offer protocol design professionals. I mentioned adaptive designs, and we're going to really get into that in a lot more detail. This has been a very, very promising area. It took a while to really get traction in the marketplace, and we started tracking adoption around 2012. We estimate that we may be looking at 40 to 50% of major and mid-sized companies now that are using some form of simple adaptation. It might be sample size re-estimation or early termination due to safety or efficacy futility. And there are a number of other adaptations that are becoming more commonly used today. Here, the definition of an adaptive design, and I think we'll get into this in more detail, is really to have pre-planned changes that have already been approved, pre-planned changes to the protocol, and while the study is underway, to, during the interim phases, review the data and actually make adjustments to the design of the protocol while the study is underway. We're starting to see a lot more momentum in the adaptive design space. The regulatory agencies are becoming far more receptive to design, and they believe it offers a lot of promise in terms of even improving success rates. And we also know that the 21st Centuries Act is really now requiring the FDA to issue more detailed guidance, which we think will create more regulatory wins to drive adaptive designs into more mainstream. We're seeing now also a lot of organic growth, the belief that a lot of our randomized controlled trials really will respond well to some of the adaptive design approaches. And we also see new applications, many of them associated with a lot of the new types of trials that are becoming more popular, many that really target very, very specific and stratified patient subpopulations, very costly studies that are targeting rare diseases where we really need to make sure that every cohort in our studies counts. So adaptive designs really help to channel and focus a lot of our resources as well. So I leave you again with the key concepts, and I also encourage you to send me a note if you'd like to receive any copies of these papers. Are we saying that protocol complexity is bad? No. Again, we believe that complexity will continue to rise. It's all about managing complexity and having more of a management and mitigation mindset. Are agencies to blame? We think that a lot of the blame really can be directed at the protocol authors themselves, but the good news is that there are many opportunities to impact and improve and optimize protocol design. When we can point the blame, we can also identify so many places where there are opportunities for practice improvement. And we've identified a number of opportunities targeting those non-core procedures, as well as avoidable amendments as critical places where there could be substantial resource savings that could really be better applied to other programs in our portfolios. So I thank you again for the opportunity to share in the celebration. Jim, do we have time for a couple questions?
Yeah, I should have mentioned that up front. We will take a few limited amount of questions now, and then we'll have an extended Q&A during the panel after all three speakers are finished.
So this is the time for core questions, not non-core.
Right.
And I'll also suggest that if you have any of the non-core questions, that it's fair game for the other panel members.
Very good presentation, Ken. I'm just wondering, in terms of the protocol amendment, do you have data by therapeutic area or major?
Yes.
Okay, so is there any, because I would be surprised if oncology would be, I will not be surprised if oncology will have more amendments versus a more established therapeutic area like the cardiovascular and metabolism.
Yeah, you're exactly right. Oncology is really quite an anomaly. They're typically one of the most complex disease conditions, and we see a much higher relative number of amendments, particularly in later stage studies.
So is the complexity more about therapeutic area?
It depends, again, on who you ask, because specialists in any disease condition can point to their own design practices and identify places where they're conducting procedures excessively, when there's a growing number of non-core activities. But if you were a company that had a diverse portfolio, you could earmark some of the most complex therapeutic areas as a place to start, and others could be sort of left for a later time period.
Ken, you mentioned that the number of patients per trial has gone down. Do you think that's a function of companies moving away from primary care more towards orphan and specialty care? Is that,
Yeah,
do you have data for that?
We think it's two factors. One is what you're describing, that there is movement towards specialty and rare diseases, which have much smaller numbers of patients, clearly. The other is that if you look at the number and types of companies today that are funding drug development activity, we've seen a huge shift away from major pharma, and we see lots of smaller companies that are targeting some of these specialty and rare diseases and also have more limited resources. And so they're trying to power their studies with a smaller number of patients as well. So those are really sort of the two factors that are quite prevalent. Today, major pharma is only sponsoring around 38 to 40% of all drugs in development, which is unheard of. Well, great. I look forward to the panel discussion. Thank you all very much. Thanks, Jim.
Thank you, Ken. Our next speaker is Joe Massaro. Will be speaking to us about designing and operating a high-performing DMC/DSMB. A little background on Joe. He's a professor in the Department of Biostatistics at Boston University School of Public Health, focusing on teaching statistical methods for clinical trials. He has over 30 years experience in the design and analysis of clinical trials, DMCs, and adaptive designs. Prior to joining BU, he was the Assistant Director of Data Management and Biostats at Quintiles, and is currently a statistical consultant at Boston Biomedical Associates and a general statistical consultant to the pharmaceutical and biotech industry. With that, I'll hand it over to Joe.
Thank you. Hi, thank you very much for coming, and thanks very much to SDC for asking me to speak here. I guess I need my slides up. Well, thank you. Okay, So what I want to talk about is data monitoring committees and kind of the interaction with them and how to, designing and operating them. We give an outline, the brief role of the DMC. We'll discuss that. Then I'll talk a little about the FDA guidance on DMCs and DMC charter. And especially focusing on the confidentiality of results of the interim analysis. And related to that, the final topic will be on how the DMC can communicate with the sponsor so as not to divulge results that may be confidential, because it does lead to operational bias. Okay. So here's the setting. This would be a randomized, controlled clinical trial. It could be double-blind, single-label, open-label. But it is a randomized design. And we'll talk about the situation where the DMC inspects unblinded interim looks of the data, so separated by treatment group. And this is at the interim, so it's prior to all patients being treated and followed. And the DMC will make recommendations based on this interim look about future trial conduct. And examples of some decisions could be simply continue the trial as planned based on the interim analysis. But there could be safety concerns. For example, stop the trial early if there seems to be evidence of harm, especially of the new investigational product. From an efficacy perspective, in terms of interim looks at the data, the adaptive design, these are just examples of a few recommendations that can be made. Stop the trial for futility of the experimental treatment. Based on the interim data, there's really no difference between the treatments. It doesn't look like there's going to be any difference between the treatments by the time the study's over. So it might be worthwhile just to stop for futility to save time, not waste patients' time, and save money. Or something more positive, stop for overwhelming efficacy of the experimental product. The experimental product is working a lot better than we thought it would based on this interim looks. So let's perhaps stop and start the approval process. Others are increase the sample size re-estimation. Based on the interim looks, things are looking good, maybe not as well as good as what you thought when the study before the study started, but still promising, but we need a larger sample to maintain adequate power. So those are just examples of a few decisions that the DMC will make based on their review of the interim unblinded data in a protocol that allows for such interim looks. Okay, but it's important that the sponsor be kept, and really anyone involved in the study conduct, be kept unaware of those results as much as possible. These are a few quotes from the FDA guidance on DMCs, on data monitoring committees, from the section titled Confidentiality of Interim Data and Analyses. And basically they say that, look, if a sponsor knows the interim data, well, first of all, it's not necessary for the sponsor to know the interim data. And in the middle bullet point, it's because it can bias the outcome of the study. If the sponsor is made aware of unblinded results, then who knows how it's going to affect the remaining conduct of the study. Okay? And the bottom bullet point there says, you know, if the sponsor is aware of the results, it may become impossible to make changes in the trial in an unbiased manner. So it's very important to keep the sponsor unaware of the results as much as possible of the interim analysis. But we'll see that sometimes that's a little easier said than done. Because the DMC does have to communicate to the sponsor or the steering committee following their review of interim data. And just what should they say? And how do they say it? Well, in the guidance it says the communication should be as terse as possible, okay? You know, study can continue as is, and that's it. I mean, you can say, hi, how you doing? You know, like, but study can continue as is. But then if that's not the recommendation, then it has to be more detail maybe needs to be given. For example, the DMC recommends increasing the sample size by 50 per group. But even then, there really should be no discussion of to why. It's like, what did the results look like that led to that decision? The DMC, the sponsor needs to either go with the recommendation or not, so that they don't, without looking for more information, which is not an easy thing to do. Okay? But there is a point where if the DMC recommends stopping the trial, if they say to the sponsor, we've looked at the interim data, we recommend stopping the trial, either because of futility or excellent results or safety. Well, at that point, it's unfair to ask the sponsor to stop the trial without themselves maybe taking a look at the data. Okay, it's difficult for a sponsor to follow a DMC recommendation in that setting. Okay, and I'll give you one example. I'll give you actually a couple of examples of the DMC, kind of where there's had to be difficult DMC communication with the sponsor. Here's an example from a safety perspective. This was on an oncology trial I was a DMC member on a few years ago. There was a certain type of cancer, and it was, you know, best medical care or placebo was the control versus the experimental. And after 40 patients were enrolled, 20 in each group, and treated for several weeks, the DMC inspected unblinded safety data. And it turned out the experimental treatment had 4 life-threatening serious adverse events, and the placebo group had none. Not only that, the 4 life-threatening events were all related to liver function. So it looked like something was going on here. Now, as the statistician, I was asked to calculate a p-value, and it turned out there actually wasn't a significant difference between groups. But at the same time, when you're talking about safety, statistics isn't as important. So the DMC did make a recommendation to terminate the study based on these 4 life-threatening liver SAEs that occurred in the experimental treatment and none in the placebo. But you know, it took about 3 or 4 hours to come to that decision. There was a lot of discussion back and forth. Okay. But the sponsor got back, so the DSMB had their meeting, and the sponsor got back on the phone, and the DSMB made their recommendation, and they had to explain why. Okay, their recommendation was to stop the study for safety, and they had to explain why. Now, it's a recommendation by default. The DMC does not have the wherewithal to stop the trial. They just make recommendations. If the sponsor did not follow the recommendation, now you have a potential issue, because now the sponsor is made aware that there are 4 experimental treatment group liver function adverse events. And will this awareness affect the subsequent study conduct, even unintentionally, okay, causing bias in the remaining safety data? I mean, what if when the trial was done, those were the only 4 liver-related serious adverse events? Would people start thinking, well, something's going on here? They changed their reporting. I mean, bias and the perception of bias are almost just as bad. So it turned out the sponsor did follow the recommendation, but that wasn't an easy, they didn't say that day, we're going to follow your recommendation. As soon they had to go back and think about it, but any repercussions about telling the sponsor the result, you know, especially given the FDA concern about confidentiality of data, had to be weighed. But the sponsor did decide to terminate at that stage. Okay. So again, for efficacy now, what information does the sponsor need to be made aware? if it's to increase the sample size, that's really all they should be made aware. Increase the sample size to X. And then again, the sponsor needs to decide whether they should follow this recommendation. Okay? They ideally need to do it without knowing the underlying reason the DMC made that recommendation. Is it fair to them to do that? Okay? And it might not be fair, but if they have any knowledge of the results that made the DSMB recommend increasing the sample size, there goes your potential confidentiality of results. There goes the FDA's concern in the guidance about sponsor knowing too much, affecting the remaining conduct of the data. I'll give you another example. Well, this example is related to efficacy. Okay, this was a rare disease, and it was very difficult to enroll. It was a randomized trial. The sponsor needed 80 experimental and 40 placebo, and the outcome was mortality. Okay. The DSMB recommended increasing the sample size to about 30. They did an interim efficacy analysis about halfway through, and they recommended increasing the sample size to about 30 subjects. And the sponsor couldn't do it because it was such a rare disease. It's going to take them, they said it's going to take us long enough to get the original 80 and 40. Okay? So the trial continued. And when it was done, I was the statistician on the project. I wasn't on the DMC, but I was the one who received the final data, and I generated the results. And when I got the final data set, the results was a significant beneficial treatment effect. And it's not often I get to spread good news like that, so I was very much looking forward to the conference call the next day with the sponsor to tell them that you had a significant beneficial treatment effect. And the response that the sponsor gave me was, how could this have happened? And they were quite upset. Now, as I sometimes have to ask my wife, did I do something wrong? Okay, I didn't know. Okay, I didn't know what to say. So I said, well, what is the problem? It turns out, after the DMC made their recommendation, the sponsor sent out a press release. And the press release said, I took a look at it afterwards, and it said, the DSMB recommended an increase of sample size of X to maintain 80% power. We cannot do that, okay? However, we are still considering the study supportive as part of our program, because they thought the DSMB recommendation that they couldn't follow meant their study was going to fail, okay? And that is the vibe you get from the press release. And they got mad because they had investors pull once that press release came out. And now, if investors find out the results were significant, would they sue? And, you know, so there's that concern, but there's also the concern of how will investigators involved in the study take this press release? Will they be less enthusiastic about participating in the study? How is that going to affect results? How does the sponsor itself affect the remainder of the study? Because they will probably seem less enthusiastic, okay? So I explained to the DSMB, I'm sorry, to the sponsor, the DSMB asked you to increase the sample size. You couldn't. That just means you didn't have 80% power for a successful result. But guess what? You still had 70% power for a successful result. So you still had a good chance of a successful result, and that chance came through under the trends that the DMC saw in the interim data. So you have a certain type of bias. The DMC did basically what they were supposed to do in terms of their communication, but the misinterpretation of the results by the sponsor, did that lead to a certain type of bias, okay? So it's important that sponsors are made aware of what these recommendations really do mean. Okay? So again, did the sponsor's feeling of a failed trial affect the remaining data after the DMC made their recommendation and the sponsor couldn't follow it? Did investigators feel the same way? Should the sponsor have even sent out a press release following the DMC recommendation? Perhaps they should have, but because people might be anticipating the DMC meeting, but what does it really need to say? You really got to sit down and think it through. And again, make sure that the sponsor is not misinterpreting results. Okay. Another quick example, I was involved in a trial on graft versus host disease, and these are the interim results. The outcome is treatment failure by day 80. And that's bad. And the placebo had a lot less treatment failure than the experimental. So much so, these were basically the exact opposite of what was anticipated in the protocol. Okay? So how does the DMC tell the sponsor this? Because the first thing the DMC thought of is, this is so bizarre that it's the exact opposite of what was anticipated in the protocol. Was there a mistake made somewhere in the randomization, for example? Did somebody mix up the randomization code? I was the one who ran the analysis, so I started breaking out in a sweat and saying, oh gosh, wasn't me, what did I do? But it turned out, all kidding aside, I checked to make sure that when I saw the results that there wasn't an issue. So what do you tell the sponsor? So the DSMB called the sponsor after inspecting the results and did not say anything directly, but said, can you double check your randomization process? Can you double check the makeup of the treatment kits and things like that? So the sponsor is kind of aware something's wrong, but the DSMB did not tell them results. So the sponsor took a couple of weeks to check, and no mistakes seemed to be made in anything, especially in the randomization. And so at that point, the DSMC said, okay, this is what we're saying. And so they had to be unblinded. The sponsor had to be unblinded. But at that stage, there was no way the study was going to be a success, even if the sponsor continued, in all likelihood. So in all likelihood, the sponsor would stop the study, which they ended up doing. But, when you're giving a sponsor unblinded results as a DMC person, you just got to really make sure all the bells and whistles are in place. Okay. Just general issues. If the DMC says increase sample size to X, and we saw one misunderstanding, what does also happen is people feel, oh, we must be close. We just need to increase the sample size a little. We could do that. We must be close. We must be close to significance. Well, will that not process bias anything? Okay. One time I was involved in a study where the DMC said continue the study as is, and I had an investigator call me up and say, well, the product must not work because the DMC said continue the study as is. If the product worked, they would have stopped the study. And I had to explain, no, you have to really have overwhelming efficacy to stop the study. But even something simple as continue the study as is can cause bias. This is one of the FDA's biggest concern. Here in the U.S., they will often require, the statistics is nice to work out the increase in type 1 error and all that. And, there's a lot of methodologies available. One of the biggest things the regulatory agencies ask for is how will you minimize operational bias entering a study? We want a plan. And big part of that plan is having an independent DMC, but outlining the communication process, etc. Okay, so just in conclusion, the DMCs make recommendations on study conduct issues while the trial is going. Confidentiality of results from the sponsor confidentiality of the results from a sponsor as much as possible is necessary to avoid bias entering the trial. DMC recommendations with the sponsor can have repercussions, need to be as terse as possible, and important to consider repercussions of communication and minimize bias. Thank you very much.
Anyone have any initial questions for Joe?
Based on your experience, post recommendation for modification to the protocol, is the FDA doing any empirical evaluations of the results based by stratifying the results to look at operational bias?
Like before and after?
Yeah.
Yes, I mean that's often requested in the study to make sure that the trend in the data before and after, that there's not a significant difference with respect to the treatment difference, if you will. Yeah.
So how do they, when that occurs, and it could occur by chance, just because the sample size is split, it could
very well be.
How do they resolve those types of situations?
I haven't been involved in a situation where it was an issue, thankfully. But you know, it could, if there's not a reasonable explanation, it could jeopardize the trial. Yeah.
Thank you, Joe.
Hi, Joe.
How you doing?
So I found your example of the sponsor's press release pretty interesting, because I think it's actually very relevant to small companies. I know that at a large company, the DMC increasing the sample size would not be a big event, and it would not be considered for a press release. But at a very small company, such as the one that I'm at, I have arguments with our lawyers about what's a material event, and our legal counsel tells me that they're obligated, being a publicly traded company, to report material events. And so that is a judgment call. And so there's a lot of education that I think the statisticians need to do to not only upper management, but investor relations and legal counsel about what something like this means for an interim analysis in the middle of a trial. And maybe that's something that the statistical field needs to consider.
Yeah, I 100% agree with everything you said. It is more, this was a relatively small company. This is their only product. Now, they had other trials ongoing on this product, but this was one of their phase 3s. And I also agree about you making sure, you know, a statistician or somebody make sure that the press release doesn't say too much. The sponsor never should have given an indication, well, we're going to consider this supportive. Like, they never should have given the vibe that doesn't mean it's successful. If anything, give the vibe this doesn't mean, you know, say something like this doesn't mean it's not going to be successful. It just means maybe, you know, I don't know how far to go. But yeah, I agree. It was the vibe in this case that the study was not going to be successful that caused the problems, I think.
Hi. Very interesting.
Thank you.
I see the potential benefit to both the patients and to the sponsors for having a data interim committee. Can you just tell me, as a sponsor, what would be the risks or benefit to do this? Because it's not mandatory to do it. But if you're a sponsor and you want to have the best possible outcome, would you recommend that this is a routine practice, or are there risks to sponsors other than what we just talked about?
I generally recommend it. I mean, and in fact, I'm on some trials I'm on where the protocol doesn't call for it. The FDA statistician will write back, consider doing an adaptive design. We actually get recommendations from the FDA. I think the biggest issue is this operational bias. If you can show that, you know, a plan beforehand that this will be minimized as much as possible. But a lot of sponsors I work with, when they come up with their sample size calculations, they're very unsure about the assumptions. And so for that reason, I generally, if they do, I generally recommend this. We just got to make sure this is all worked out as much as possible. Yeah.
Did you employ the zones, like the futility zones?
Exactly. I mean, so you lay out, you have to lay out in the protocol that if the conditional power, you know, is between 35 and 80%, you're going to increase the sample size. You know, and that's the other concern people have is this, what about this back calculating? You know, could somebody back calculate what the sample size recommendation was based on? And then if it's less than 38% or greater than 80, there's no change to the protocol. So if there's no change, it could mean basic failure or close to failure or tremendous success. They don't know. But they will, you know, they do have some idea that, some, you know, especially when it's increased the sample size to XXX. Okay. All right. Thank you very much.
Thank you, Joe. right, our final speaker, last but not least, our president of SDC, Dale Usner. He'll be speaking on adaptive design, practical considerations, and execution. I've had the pleasure of working with Dale over the past 9 years, first as a client, and over the last 6 years as a colleague. We've probably collectively flown a couple of million miles together in those past 6 years. But one of the many great things about working with Dale is he's able to help people understand the importance, impact, and implications of items from a statistical perspective, regardless of that individual's role within the study team. Prior to joining SDC, Dale's experience included leading stats and data management teams at sponsor companies, AVI Biopharma, Cymogenetics, and Bausch & Lomb. Also, large CRO experience with PPD prior to that, as well as statistician here locally at NERI. Dale has an extensive array of therapeutic experience, probably easier to list areas that he hasn't touched at this point than what he has, and a lot of extensive experience as well interacting with regulatory agencies on behalf of clients, and prior to being at SDC on behalf of his sponsor companies. So with that, I will turn it over to Dale.
Thank you very much again. Thank you, Jim. Thank you, Ken and Joe. Everybody, what I'm going to be going through now is sort of broadening, hopefully, people's minds on what adaptive designs are outside of increasing sample size or stopping for futility, but doing some other adjustments that probably will add to the complexity that Ken was mentioning, at least on the short run, hopefully reducing the complexity in the long run. And all right, so the key two references I used for this were the two guidance documents from the FDA, one from the drugs division and one from the device division. The device division is final, the drugs division is draft for 7 years. They were pretty similar, so I think they used each other. My agenda, I'll be going over the definition, some of which was already discussed, motivations, disadvantages of adaptive design, what you're able to modify or what's currently being discussed as possibilities of modifications in trials, interim data and what you'll do with it, candidate studies, so where is it good to possibly have an adaptive design versus not, examples, and then questions and answers. And I don't stand well behind this, so if I'm in front of somebody, tell me to move. Definition of an adaptive design clinical trial, prospectively planned. You've heard that before, you're hearing it again for a reason. It must be prospectively planned so that you are able to essentially keep the bottom bullet designed to maintain study integrity and scientific validity. It's an opportunity to modify one or more aspects of the study design, study conduct, hypotheses, or analyses. You can build an adaptive design to change your primary endpoint. You analyze interim data from subjects in the study, not external to the study. It can be blinded or unblinded. It can be with or without formal statistical hypothesis testing. So one example of an adaptive design, it's I don't consider it adaptive, but everybody else does. Changing your inclusion or exclusion criteria mid-study for one of multiple reasons. It could be that you are expecting a certain broad basic characteristics in your patient population and you're not seeing it. And so you're having to modify something in your inclusion-exclusion criteria in order to broaden that base. It could be the fact that you're not enrolling at the speed at which you would like, and so you're going to look through your criteria to determine which ones folks are failing at and ones that you're able to modify in order to improve that success rate. That's what you'll target for adaptation. those are in a blinded fashion and really would have little impact on the statistical validity to most extents. Motivation, more efficient studies, possibly shorter duration, possibly fewer patients. Can use it to drop ineffective arms, doses, or subgroups of patients. You increase the likelihood of success on your study objective by taking a look at the interim data. Do you need to enroll more patients? Do you need to actually target a separate subpopulation in your patient group in order to move forward and progress in a more efficient manner? And you're able to get more information. You're able to look broadly at the beginning, so at a broader set of, let's say, dose information or a broader set of enrollment criteria, and then based off the interim, you're able to focus that criteria. Again, this is all based off of pre-planned modifications, though, so you're going to have to take time up front to lay out what those possible decisions are going to be and understand what the impact has on your statistical rigor. Disadvantages, the key thing here, and you've heard this throughout, is operational bias. I attempt later on to go through and identify some operational biases, and it's really tough to. I think Ken and Joe hit on better ones than I, but I will also target some. Essentially, the bias is an estimate of treatment effect that deviates from its true value based off of something that is going on in the study. Knowing results of various decisions, even if it's only the decision. So if it's a decision based off of endpoints, subjects, sample size, that lead to a change in the conduct or subjective decision making. So for instance, in an area where you're possibly switching the primary endpoints in the middle of the study, now all of a sudden the investigator, if they know that you're switching that, first the question is, do they have to know? But if they know that you're switching that, how does that impact how they're actually taking the measure and recording that measure? There's going to be some bias involved. Inflation of a type 1 error. This is maintained in adaptive design, and that's why the whole prospective planning comes into place. You want to prospectively plan for all the possible decisions that you're going to make to ensure that that type 1 error, or the error of saying, all right, this product is better than the control or non-inferior to the control, that probability stays at a two-sided alpha over 5 in general. And it requires simulations for complex design characteristics. There really aren't programs out there that you can just press a couple buttons and it'll tell you. So there's more effort at the design stage based off of everything I just said. Frequently difficult to interpret if it's an overly complex. So you really want to target 1 or 2 modifications at most. If you're starting to target 4, 5, 6 modifications, your statistician might have some fun in the short run, but at the end of it, trying to actually interpret the data will be very difficult. And it can also increase the sample size and duration just because of the number of looks in the alpha adjustments. But it also allows you to understand your data midstream. Possible modifications are to study eligibility criteria, the randomization procedure. Do I want to mid-study I take a look at the information and I see I've got a lot more variability in one of the treatment groups than the other. I've known that that's possibly going to be the case, so I've set up a modification to change from a 1-to-1 randomization to a 2-to-1 randomization in that case. Treatment regimens. You can think of basically a dose response situation where you're dropping a dose in the mid-study. If you're not quite sure what doses to go into your phase 3 with or your pivotal trials with, so you go in with 2 or 3 doses, take a look at the data, again with appropriate designs, and make decisions to drop a dose regimen. Total sample size has been discussed, schedule of patient evaluations, primary endpoints, the order of secondary endpoints. If that order is, a lot of times there's a lot of secondary endpoints, really difficult to order, how you're going to put that up front as far as maintaining alpha and really trying to select that right order. The interim analysis can help with that on an adaptive design, and analytical methods of endpoints. Types of data that can be used, I'm going to go through this pretty quickly. Basically, demographic baseline characteristics, I gave a brief example of that already. Safety outcomes, pharmacodynamic or biomarkers, as well as efficacy outcomes. And again, this can be blinded or unblinded, but the reason I put this up here is, as a statistician, I always get the request of, well, as long as I only call them treatment A and treatment B, that's still blinded, right? It's not blinded. That is unblinded information. So anytime you go to that level of information in order to make a decision, that is considered unblinding. Candidate studies, pretty standard. You've probably heard this before. The key thing is timing of endpoint that you're going to be making decisions based off of relative to enrollment enabling adaptations. So if you've got an endpoint that you're looking at that's 2 years out and you're able to enroll in 2 months, the full study, you're not really a candidate for an adaptive design. Or if you have a shorter term endpoint that's predictive of the primary endpoint, once again, few targeted modifications. So if you're finding yourself in a situation where there are many unknowns, definitely would suggest another phase 2 or early phase trial that's more exploratory to get more information on those unknowns. And then finally, if your samples, this doesn't account for everything, but if your sample size is driven by safety and you're trying to reduce your sample size, then it might not be a meaningful way to use your time for the adaptive, because you have to have the sample size to drive your safety profile anyway. All right. I'm going to go into examples. Am I speaking at a good speed? All right. First example I have is a sample size reassessment study of ADHD, a randomized 1-to-1 test to control. The primary endpoint is a physician-rated ADHD scale. I specifically picked this endpoint because it's very subjective. The considered modifications are sample size reassessment, and that the separate decisions by gender. For some reason, there's a known aspect of this drug that it possibly works different by the 2 genders, and so that the sample size reestimation decisions are by gender. So this next bullet, the design properties and varying scenarios, I've removed that from the slides from here forward, but please understand that it's there. It's for space reason. For every one of these, we would need to walk through the scenarios that are possible in these modifications, determine decision rules, and then understand from those decision rules expected sample size, expected duration, maintaining the type 1 error and calculating the type 2 error. And in that way, you're able to take a look at the adaptive design that you've set up. You can compare it to conventional design. You compare it to other adaptive designs to see if it's going to give you benefit to run this adaptive design. And that's a lot more difficult than 2 bullets, but that's what we'll go through here today. The key things that I really want to discuss, again, are operational impact and bias. So there's a couple of things here. Let's consider that when you did the sample size reestimation, you require significantly more females than males. And so I have this next bullet. I didn't quite know where to put it, so I put it there. But let's talk about that on the requiring significantly more females. And as discussed earlier, if that gets back to the investigator or if that gets back to the sponsor, there's possibly going to be some operational bias injected because now they're going to intuit that, all right, this isn't as effective in females. And so what is that going to do to the measures that are taken? And one of the key things that I've been asked to do, and it was brought up in the question and answer, and that is analyze the data pre and post interim to see, all right, is there an effect by stage? Thankfully, again, I as well have not come into a situation where there's been a difference and that I've had to justify, though I will talk about another scenario right after this where I can believe there would be, and so I wouldn't recommend it. So some of the other questions on the operational impact that I get frequently are, should we stop enrollment during the interim or continue on? And that, to me, really depends on what your enrollment rate is, what the cost of the patient is, what the cost of stopping is, and restarting a site, because it's not free to stop and then restart. To get the sites re-energized and going back, it takes a lot of time as well. So that has to be a discussion. And the key thing here for me is the different sites enrolling pre to post interim. I've had this a lot on adaptive designs or where there's an interim for sample size re-estimation where for some reason, folks will want to focus on their key sites right away. Maybe it's just the first sites that start up. It's their primary sites. They go in, they get those started up. Let's say they're US sites. And then they'll enroll on that, make the decision, while they're opening rest of world. And now rest of world is what's enrolling the next set of patients. And so you can see there that that's going to inherently possibly add bias because we're in two separate areas now of the world. And so I would caution you to strongly think about that as well. And I see it in phase 2 to phase 3s where folks will target their phase 2s on a couple of key investigators, but then want to take that information and develop their phase 3s. And they're not going to have the same variability frequently in those phase 2s because of those investigators that they targeted and because they're targeting only 1 or 2 instead of the 20, 30 that they see in phase 3s. So my next example is a dose pruning, and this is more of an example of some pre-thought to designing the study. And so when people think of adaptive design, they look at the statistician. I, as a statistician, like to look at everybody else because it's not just us. We'll do the mathematical part, but there's a lot of operations that also has to be accounted for, and that's really what I'm trying to discuss here. So if we have an example of 3 active doses and 1 placebo, that can also be 2 active and 1 placebo. The number 3 isn't magic. We just don't know what doses we want to go the whole way through our phase 3 with, but we want a phase 3 now. And so the considered modifications would be to discontinue non-efficacious doses and possibly modify the randomization, which is what I mentioned earlier, from a 1-to-1 to a 3-to-2. And so the operational impact, after doing the impact that I described earlier about looking at the different scenarios of type 1 error, type 2 error, et cetera, operational impact, are we using an IRT system? Are we set up in that IRT system now for the possible modifications? Because the last thing you want to do is make a modification and now have to go into your randomization system, update it, and retest it all to make sure it's working correctly. Pre-build that into your system. And I've done that, and it works very well. You get to test everything right up front. It really helps from the beginning. So these are just some things to think about. Manufacturing, kit product and availability. You're going to have more call for some of, if they're different kits essentially, or for the different doses, you're going to have more call for the doses that remain on study. Make sure that you've got that available. And then also on the shipping side as well, that, I jumped across resupply algorithms. They would possibly change as well, and then shipping. All right. Here's an example of a primary endpoint where I'm looking at a study of dry eye, and I've got hierarchical endpoints where I'm going to test corneal staining first, and if that's significant, I'm going to test the dryness symptom. Both of these are valuable endpoints. I could go multiple different ways here. I could go co-primary, where I'm doing a Bonferroni correction, and I'm going to claim the study is a success if either of those two meet. I could go higher, but that's going to reduce the alpha, cause a bit of an increase in sample size. There are other also outside of Bonferroni. I could go a hierarchical endpoint where I'm going to pre-state corneal staining dry and then dryness, but I'm not sure. All right, is that really the way I want to go? The data maybe on earlier phase studies really weren't clear enough, but this was a general direction. I can have an adaptive design set up front where my considered modifications are order of hierarchy and the sample size reassessment. So you're going in and taking a look at that hierarchy at the interim where you might have a lot more information than you've had from your prior studies, make a decision and move forward. Operational bias here, concerns, if, all right, all of a sudden you were, going corneal staining as primary and then dry eye or the dryness symptom is secondary and now you switch if the investigators know about that or others, what is the impact that that's going to have on their assessments of these endpoints? I can't tell you. That's the concern. So must the investigator know at this point that this decision has been made to switch? And I would argue they do not need to know in order to basically maintain status quo and to not interject operational bias. You can make that decision and not pass that forward to the investigators. It's not meaningful to them in their assessments. All right. Okay, and then lastly on the composite endpoint, I was trying to come up with a couple of examples for different areas. This one is really to ensure collection of the data on the EDC system up front, that you account for any possible modification in the study based off of an adaptive design, that what impact that might have on the EDC system. I also threw in here risk-based monitoring, so let's walk through it. So if we consider our primary endpoint as a composite endpoint, but we have an adaptive design that actually allows us to change what assessments go into that composition. Pre-stated, whatever they are, we do the assessment of alpha, power on the stat side. Operational impact questions, I'm assuming all the measures that are possibly going to be modified to in the composite are already collected in the EDC. You need to have that to assess whether or not it makes more sense to change out those individual assessments in the composition. What about the risk-based or targeted monitoring impact on this, possibly on an adaptive design? I've seen some areas where targeted monitoring, they're focused on the primary endpoints. That's what they're going in and source data verifying, as well as a random assessment of other things. Now you're going to, midway through the study, change what's possibly going into that composite endpoint. So it's something to think about with respect to how do you build that system in order to change over midstream, because you don't want to be doing those types of changes mid-study. Pre-build that. So this is, I'm really trying to understand, basically point out that while this is statistics heavy, it also has a lot of operational impact. And then operational bias here, of course, pretty similar to what I've been discussing. If investigators are knowing that you're pulling out a certain assessment from the composite, then they're probably going to be thinking, all right, we're really not seeing efficacy there, but we are really seeing efficacy on these other assessments. That's why they're putting them in. What does that do to their assessing of those endpoints? This is my last slide. Sorry if I went. This is the seamless phase 2-3. This is, I just put this on because this is what everybody frequently thinks about on adaptation. And I would just really caution that, a lot of times when this is asked for, there's more than one or two modifications that are being thought about. There's a lot of not understood information about the product or the product in the indication. So you really just have to weigh, are there really only a few things that I want to understand and possibly modify versus do I need to run a phase 2? Take the time to truly analyze that data from a safety perspective and an efficacy subgroup to get a full understanding of that data. That's it. Thank you.
I actually wanted to understand, I guess you had very good examples about changing the primary endpoint, right? So there is a big regulatory risk by changing the, if you go to the FDA, talk to them, they hate it, right? Because we have been there several times about it, and first of all, so I'm just wondering, how did your regulatory interactions go, number one, and we have not seen a single scenario where they have approved changing the combination of a primary endpoint, because there is an inherent question about how are you going to do your predictive power, how are you planning to do your conditional power, and if you're trying to make a decision based on a primary endpoint on a conditional power based on one endpoint, what's the second endpoint looking like? Especially if it's a pivotal trial, it comes to a point that the pivotal endpoint is actually regulatory approved endpoint. So so I'm just very interested to know about the regulatory interactions, because my experience is we have horribly failed.
Yeah. So they're definitely challenging, for sure. And I think I've possibly been in scenarios where the primary endpoint hasn't been as well defined, and they are, because of that, a little bit more open to hearing the adjustment. The composite, I have not done this one. This is an example from one of the guidances. Well, I created the example, but the composite endpoint itself was an example from the device guidance straight up.
We're talking about adaptive design and then possible changes. And I think what's throwing me in my head is that in my mind, when I think you're running an adaptive trial, you're thinking ahead of what you could do. Just because you make a change, whether it be a result of a DMC oversight or whatever, does that automatically make it an adaptive trial? Like, I'm a little confused there. It's like almost like we're mixing the terms.
Okay, I apologize. I'm definitely missing your question.
Okay.
So the pre-planning on the adaptive design is key in order to really understand the working characteristics of the underlying statistics. So are you asking?
Just making a change in your study doesn't make it an adaptive trial. It's showing that you've made a change.
And depending on what that change is and what information you had when making that change could make it a study that's possibly not worth continuing because it's not going to be looked at in a positive fashion. So
but, I mean, you had said earlier that you didn't think a change in inclusion and exclusion criteria was an adaptive. And I agree. I mean, to me, it's showing you that there's some parameter of the conduct of your trial or your criteria that you need to change. But in my mind, when we say we're going to have an adaptive trial, your protocol is going to state, after enrolling the first 100 patients, if we don't see certain criteria met, we will, lessen it or, something like that.
No, so I am in complete agreement, and that's actually why I threw out that example and said I would not have recognized it as such. The guidances consider that adaptive. So yeah, and
so
I
you're
think
adapting to whatever is.
Sorry.
No, you're adapting to either the safety profile, the efficacy profile, or operational profile.
Exactly it. So it doesn't have to be on efficacy data, as I had in one place. It doesn't have to have any formal stats. You're basically taking a look at the information in the study, so it has subjects in the study, and making a change in that with that information. Yeah, absolutely.
Can you comment on the advantage versus disadvantage ratio for running an adaptive design for a superiority trial, let's say versus placebo, versus a non-inferiority? Is there anything in these two basic structures that say, better adaptive or not? Based on whether it's in a non-inferiority or a superiority trial, all other things being equal?
Yeah, that's a really good question, because I do that in pretty much everything else I do in statistics, and I haven't thought about it in this situation. Yeah, I think the only thing I would possibly caution against or think to in that situation is that operational biases that might somehow lead to overstating efficacy. I would have to look at how the operational biases impact the primary endpoint with respect to, are you going to be more likely to claim non-inferiority because of the operational biases, or less likely, and how that compares to superiority. And I will think about that and get back to you. I apologize, I do not have an answer.
Thanks.
Yeah.
Thanks for your talk. I enjoyed it. Could you give us an example of where the seamless phase 2-3, where you would recommend it and where it would have clear advantages?
Yeah, I personally, so if you have a lot of information on the product, so I would say in general I personally don't recommend it because I think people try to do it as a way to shorten their time, and what ends up happening is while they find out some information on the phase 2, they are more likely to actually have to change the phase 3 aspect of it mid-study. So if the product is well known, your serious adverse events, your safety profile is well known, and possibly a similar indication, you also have good information on the efficacy with the indication that you're talking, and you're just maybe looking at a focused set of dose ranges in the phase 2 from what you've done previously, just to make a decision on what dose ranges to move forward with in the phase 3. So I would recommend it in scenarios where you have a good amount of information on the product, possibly in the indication, and also have only a couple of unanswered questions.
Dale, when you mentioned statistical analysis, is it possible to take an adaptive design where you check to see whether there's baseline imbalances between treatment groups and then change the analysis to a change from baseline, or or an regression analysis with the covariates. Are those acceptable?
So technically, per the guidance, they do mention similar to that, though, as mentioned previously on another, I've not personally been successful in this situation where I'm looking at an interim information and then basically designing it to adjust for covariates based off of that. But that might be division specific as well.
All right, thank you, Dale.
Thank you very much, presenters. Thank you so much. Thank you all as well for joining us. And have a great evening and safe ride home.
Thank you.